Hello, Dr. Mark. Welcome to the Fogsi podcast series. I'm so glad that you joined and you are giving your valuable time. Now, the Foxy organization, as you know, of 286 societies, Thamunzagadi, 286 societies with 46,000 gynecologists, and we have 30 committee chairpersons. So every night, so if it is a 29th, it will be Dr. Anu who will be coming on 29th, and every day is been given to one chairperson. They come out with the faculty of academic, non-academic background, spiritual, philosophical, and we share a 45 minutes dialogue to them. I just joined to say hello to you all, and I will hand over to Dr. Anu to take further proceeding. So sometimes definitely we'll meet and we'll see. Probably I'm coming on 11th because I'm getting my fellowship by Royal College. So you happen to be around, I will be glad to meet you in person.
I'd be very happy to meet you, and thank you for the honor
Thank you
of
so
being in
much.
your program.
Thank you
And
very
Dr.
much.
Anu, you can take over from here.
Thank you very much, Dr. Tandulwadkar. And this series has been going on with great success. I welcome everybody, all the audience who have taken time to join us tonight. And we have a very special guest tonight. And I first start by thanking Foxy, all the office bearers, Dr. Sunita Tandulwadkar, to lead the team and create this beautiful platform. Dr. Tandulwadkar herself is a very leading obstetrician and reproductive medicine specialist and a top endoscopic surgeon based out of Pune, but very globally acknowledged and trained a lot of people. That is, I think, I must mention, she has trained dozens of very like copies of herself. So coming to yourself, now I am very happy, very privileged to introduce you all to Professor Mark Johnson. He's leading, he's actually on a very senior post as a lead consultant of the research department in the Chelsea Westminster Hospital and also in the Imperial Medical College in London. And I was actually connected to him through very ace gynecologists, group of them themselves. And the first meeting I had with Prof actually was very intriguing in the sense that we were talking about gynecology, but I was blown away with the level of medicine knowledge, the basic science, medicine, cardiology knowledge that he had. At the time when I met him, my dad was unwell due to cardiology, and I was very fascinated about his background, and I asked some of his students, and then I was very touched by the whole profile that he has and all the avenues that he has worked with. He's primarily trained as an endocrinologist and then shifted to gynecology. So depth of knowledge is something very profound that I have understood about Prof. Very importantly, as we know, I heard him saying in one of the interviews that to be born is the most difficult thing that we ever do, which is so correct. And what he has created is he has created a charity, a very successful charity called Born. And one of the patrons of the charity is Princess Beatrice and a very active group of people involved, raising a lot of funds to help and doing a lot of path-breaking work, shaping, I would say, the future. So I would now initiate to say, I would request you, Mark, to say a few words before Dr. Sunita logs off. Yeah.
Well, firstly, I'd love to thank Foxy and Dr. Sunita for inviting me to this podcast. It's a great pleasure to be here. And thank you for the very kind introduction, which I have to say is hugely exaggerating my abilities and knowledge. But it's very kind of you to give me such a kind introduction. What can I tell you about Born? Born is close to my heart. It's about preterm birth. I think it's close to every obstetrician's heart because we hate delivering preterm babies. we can do nothing about it. And saying to mothers, parents, couples that we can do nothing to stop the preterm birth is soul destroying. And it's for that reason, a few others, but that reason most often, that I founded Born back in 2013 to try and raise funds, raise awareness, and get more bright people to come and work in preterm birth, where we
Thank
have
you,
very
Mark.
few
That
people
is,
working.
thank you, Mark. That's a fantastic thinking and organization. And just to let you know that I also in 2019 started Sponsor a Birth. It's a completely different. Sponsor a Birth is those underprivileged infertile couple who can't afford IVF. In India, IVF is not available freely in government colleges or by insurance companies. So those underprivileged people, we sponsor their IVF cycle, which is called Sponsor a Birth. Lovely to know that you are doing one organization with preterm birth. Fantastic, fantastic.
So
So our pattern is,
you did
yes.
the same when I was changing from endocrinology to obstetrics and gynecology. I set up the IVF unit at the Chelsea and Westminster, and I founded it as a cost price IVF treatment because there's a lot of privately available, and it cost a lot of money. So we, I think, charged £625 a cycle, and we charge the cost price for drugs for the same exactly the same reasons as yourself, that people cannot afford the huge prices they have to pay privately. But yes,
I
at
agree
one point,
totally.
yeah, and at one point, my clinical director said, "No, you have to make a profit," and I said, "No, I'm not making a profit," and so we had a big
No.
old.
Absolutely. And but here in Sponsor a Birth, we are doing everything free to them, including the medicine and include because they just can't afford it. And I definitely feel that only because you belong to the lower socioeconomical class doesn't mean you can't be a mother. It's a very painful thing, and that's how 2019 we started. I'm happy to know, and I always have a deep respect for people who, after achieving everything, wants to do something for the society, for the unfortunate people in whichever part. So lovely to know about it, and I'll be joining from the Viewers League, and I'll be listening to both of you. Thank you once
Thank you.
again for coming on our platform.
A great
Thank
pleasure.
you.
Thank you for having me.
Thank
Thank
you.
you, Dr. Tandulwadkar.
Thank
So we
Thank you.
kickstart from here, if you're ready. So I'll be asking you some questions.
Of course.
Um, I would first begin by asking you, like stepping back into your career many years, and what inspired your journey from general medicine through endocrinology to focus deeply then in obstetrics and preterm birth? And we'd love to hear about what were the defining moments that steered you into this line.
Mm. It's an excellent question. So I did medicine for 10 years, and I was offered a consultant post in medicine. So I got all the way to the top of being a consultant, accredited and everything. And I did a PhD in a hormone called relaxin, which probably you would have heard of, but it's not as important as in humans as it is in, for example, the mouse or rat. If you don't have relaxin in a mouse and a rat, then your labor, which would normally last about 2 hours, and you would have your 6 to 10 pups in that 2 hour period, it will go on for 18 hours, and you will have 50% retained pups. So in essence, the absence of relaxin in rats and mice makes them into humans with a much longer labor and the need to have a cesarean section to deliver their pups if you're going to have a successful birth. So I thought relaxin was the low levels of relaxin in humans was the reason that we had such a difficult labor and that our labors went on for so long. And in my head, the rationale was that, okay, in the human, of course, we're on both on 2 feet, whereas obviously rats and mice are on 4 feet. And so the pressure on the cervix is much less. So we have to have low levels of relaxin through the pregnancy in order to be able to keep the pregnancy going. And because with high levels of relaxin, our joints would really literally relax and we wouldn't be able to walk. So I had this idea that I was going to give women relaxin to induce labor. And so I spent a lot of time as a medical senior registrar actually on the labor ward. And I knew as a medical student, I really enjoyed obstetrics, but I have to say, I wasn't the best student of gynecology, but I loved doing obstetrics, delivering babies. And when I went back to the labor ward, I really enjoyed my time on the labor ward. And professor of obstetrics said to me, look, Mark, why don't you retrain in obstetrics and you could do obstetric medicine? So I thought, well, that was a good idea. So I said, okay, I'll do some obstetrics for maybe a year and a half, then at least I'll know what I'm talking about. And then at the end of the year and a half, he said, well, you could do a little bit longer, maybe another 2 or 3 years, and then you could be a consultant in obstetrics. And I thought, okay, fine. So I then converted all the way to obstetrics and did continue doing obstetric medicine, but really became primarily an obstetrician, working with other physicians who were specialized, for example, in cardiology or in diabetes. Though, although my background in endocrinology, of course, is hugely useful in pregnancy, which is all about hormones.
Absolutely, absolutely. Very fascinating. So you were slowly treading into this, but actually your
Yes.
interest actually pulled you inside this branch. So what led you to found this charity? Very successful charity, very active. I see a lot of pictures coming all out of the events that you do. And what is the mission? So please tell us more about
Our
it.
mission is threefold, really. Obviously, we really don't understand pregnancy. We don't understand why we go into labor at term. We don't understand why we have preterm labor. And when you consider pregnancy is something that all of us, every single one of us on the planet has been through, either we've all been born. So how can it be that we understand so little? And that thought, taken along with the fact that as an obstetrician, I was looking after women coming in preterm labor, and the frustration literally of not understanding why this was happening, and the inability to stop this preterm labor from happening, really made me say, we've got to do something about it. And I think in 2013, I founded Born, and the ideas in my head were to get brighter people to come into it, to get more trainees to go into preterm birth to do fundamental research so we could understand more. And once we understood more, to actually then find effective treatments for preterm labor. And as time went on, I mean, we worked and worked. And honestly, truly, as you know, and as we all know, we have made no progress in preterm labor. The most recent drug we use, atosiban, is borderline effective. In some studies, marginally better than others, but yeah, it has some effect. But one drug to treat the myriad causes of preterm labor is never going to work. And until we can identify what's driving the preterm labor and find a specific treatment for each cause, we're never going to be able to manage preterm labor effectively. So what I suppose, a moment of Damascene moment, like St. Paul walking along the road to Damascus, and he realized we had to do it differently. And so I asked all the leading experts in the world to come and work with us at Bourne and actually tell Bourne what research to fund. Because if we look around us, we have the MRC and they do fund preterm birth research. We have well-being, we have other Action Medical Research who fund single projects. And people work for a little while in an area and then they leave it. And then another group will work in another area. Nobody really has an overview of what's happening. Nobody's saying, okay, fine. So we ask this question, we get this answer. The next step is to ask the next question. So what we asked all the world experts to come together and advise Bourne which research we should be doing. And that's why we started doing the Bourne Uterine Mapping Project, which is what it says. We wanted to look at women during pregnancy as they went into labor and as they delivered to try and understand where labor started. Was it in the myometrium? Was it in the fetal membranes? Was it in the fetus? Was it in the mother? Where did it start? And why did it start? And we focused initially on term labor because we reasoned if we understood what was happening normally, then we might be able to understand what was happening when preterm labor started. And we had the vision of understanding normal labor and then using exactly the same techniques which we had proven to work in term labor, in preterm labor. And that's where we're at the moment. So BUMP is now morphing into Big Bump, and Big Bump is all about preterm labor, getting samples from the myometrium, fetal membranes, placenta, maternal blood, and fetal blood, cord blood, and trying to understand how the system works, both in term labor and preterm labor. Only then will we be able to find treatments. And we also had the insight that, okay, I'm working in London. our preterm birth rate 6 to 7%, as you know. In some parts of the world, the rates of preterm birth are much, much higher. In Ludhiana, I understand the rates are around 22 to 24% preterm birth rates, which is huge and a massive, massive problem in that area. So we felt that we should be looking and studying preterm birth across the globe so that we could understand what were the similarities, what were the differences, and if we designed a treatment, would it work both in India, in Africa, in the United States? Would it work just as well in different places as it works here, for example? So really the vision is understand preterm birth, design specific treatments, trial those treatments in different locations, and try and find a way to reduce the risk of preterm birth.
Wow, these are very basic questions, but nobody has addressed them with the depth that it needs. It asks for that
Exactly.
level of continuous dedication and time, which you rightly mentioned, that is slightly difficult in the system that we are in. And very innovative researchers have missed addressing this so far, very basic question that we all have to face. So could you walk through the BUMP initiative? Creating an open access cell atlas of the uterus that you spoke about and its significance. You've said a little bit, but would you be able to elaborate exactly a
Yes,
little bit more about this?
with
Because this
pleasure.
sounds very new to many of us. Yeah.
So we've heard recently of this sort of molecular revolution whereby we can study single cells to understand what's going on in each cell. And so we thought, well, this is fantastic. We could apply this to the womb, fetal membranes, placenta, so on, and look what each cell in those tissues is doing as you transition through into labor. And so we used single cell sequencing, but we came across a problem. If you look at the myometrium, for example, and in the placenta, the cells are huge, and the single cell sequencing is really designed for blood and relatively small cells, and cells of 200 microns just won't fit through the apparatus. And so we looked at another technique, which is called single nuclear seq. So that allows us to take frozen tissue, extract the nuclei, and look and see which genes are being expressed at a precise point in time. And so we decided that it's much easier to use single nuclear seq. You don't have to do a whole lot of processing in the day, and to understand what's going on at the cellular level. At the same time, what we did is use spatial transcriptomics. And this means we actually take a section of tissue, and you can see how a gene, the expression of a gene changes as you go through the tissue. So say, for example, as we feel, there is an inflammatory stimulus, or I should say NF-kappa B stimulus in the decidua, which influences the gene expression in the myometrium. You can see how the NF-kappa B effect goes into the myometrium and then gets less and less as you get further and further away from the decidua. So you can start to understand what is driving the onset of labor and what are the precise mechanisms and which cell is talking to which cell and what are they saying to one another. And it's really interesting to see what is going on, to be able to understand how we can then use this information to control the onset of labor, stop preterm labor, and even start term labor, which, as we all know, induction is a very slow progress, it's a very slow process, and not always successful. So hopefully the information we're going to generate will tell us a huge amount and help us both to stop preterm labor and start labor when we want to.
Wonderful.
Don't know
I'm
if that's clear
fascinated by
enough.
the lab. Yes, fascinated by the knowledge, and we are very ready for that. So, your team has explored the combination of progesterone with the aminophylline for
Yes.
delaying the preterm labor. What did early results say?
So, well, we started, it was the original, I don't know, observation back, I can't even remember when it was, by Gyan Pravah, who looked at decidualization of the endometrium, which is something much closer to your heart than mine, which is all about how do you make the endometrium more receptive. So we give, as you know, progesterone to make the endometrium receptive. But if you give the combination of progesterone cyclic AMP, you make the endometrium much more receptive. And it is this interaction between cyclic AMP and progesterone that we see, for example, in the management of asthma. We give salbutamol, which is a beta-2 agonist, which works via cyclic AMP, and we give steroids. Some people become refractory to salbutamol, but need to have steroids in order to almost reboot their receptivity of the airways so that you can respond again to salbutamol. So interactions between steroids and cyclic AMP are happening all through our body. And what we asked was, could there be an interaction with progesterone and cyclic AMP in the myometrium? We know, for example, cyclic AMP really important because we used to give beta agonists to stop preterm labor. And if somebody is responding too much to prostaglandins, we give another dose of beta agonist to stop the uterus contracting. So we know cyclic AMP works to stop contractions. But what the problem we discovered using these drugs for preterm labor was with time, they lost their efficacy, so that with time, they just no longer work. The contractions would start again. So we started to look at the way progesterone and cyclic AMP interacted, first in cells, then in tissue, and then in the mouse. And we showed that the addition of cyclic AMP to progesterone enhanced progesterone's ability to repress genes which promote the onset of labor, both in cells, in tissues, and in the mouse, and that we could delay LPS, or an inflammatory model of preterm labor in the mouse, by using the combination of progesterone and aminophylline. People remember aminophylline. It's quite an old management that we used to use for asthma back in the day. And it's a phosphodiesterase. It's like your cup of coffee in the morning. It inhibits the metabolism of cyclic AMP. And it's why we get a little bit shaky when we have coffee, and it's why I don't have coffee before I operate, so that I can have a nice straight line when I do my cesarean sections. But that tremor is an effect of caffeine, and it is like a beta agonist. It's like you're having an adrenaline rush. It's increasing cyclic AMP, and that's what drives it. So we used aminophylline and progesterone in women at high risk of preterm birth. Our question was, would they tolerate taking aminophylline? Because it causes an upset stomach in quite a few people. But the ladies were very happy to take it. We had high compliance throughout, more than 95%. So people didn't stop taking the medication. And in our study, we had 2 people who delivered preterm with progesterone alone, and 1 person who delivered preterm with aminophylline alone. It was a small study, 70 people, just to see if we could do it. The next step is to do the large multi-center randomized study, which costs a huge amount of money. So we're trying to raise money to do that study as we speak.
Wonderful, wonderful. And the way you've thought through the whole thing and taken us through this is very simple, but I wonder sometimes why somebody never thought this decades back, but there's time for everything.
No, they have. The
So,
aminophylline was used to stop preterm labor back in the 50s. And
okay.
they found 2 things, which is really important. One was that aminophylline was effective, to stop preterm labor, but also it was as effective as giving antenatal steroids in terms of maturing the baby's lung. And if you looked at the rates of intracerebral bleeding in a preterm babe, it was lower. So intraventricular hemorrhage was lower in aminophylline-treated babies, or aminophylline-treated mothers' babies, if you see what I mean, than just with steroids. So in my mind, aminophylline may have 2 effects. 1, stopping preterm labor, but 2, protecting the baby from the adverse effects of preterm birth.
I want to ask some very off the beat question right now, if you allow me, which is that does it mean that cultures which have so much of high exposure to aminophylline as compared to some other cultures, like we have a lot of tea, so some cultures which regularly intake a lot of aminophylline, would that be a confounding factor in these studies also?
I mean,
Would
it could
be
be, but
very
you, you
interesting.
you remember that pregnant women are only advised to have 2 cups of coffee a day. And that was based on some really very weak research, which said they looked at 100 women who miscarried, 100 women who didn't. And the women who miscarried all drank coffee because they obviously didn't have the hormones of pregnancy making them feel sick, because their hormones were lower because they're going to miscarry. And on that basis, pregnant women are advised not to have coffee. So we don't really have those data. And every now and then people say, oh, well, coffee could cause a smaller baby, but then nobody can really prove that. So,
Yeah.
and we didn't find any effect on birth weight in our study of taking aminophylline. So I don't think we know the answer to your question because
Yeah.
most pregnant women stop taking coffee.
Yeah, correct. So, I'll move to some little more detail about so what are the promising biomarkers or early predictors of preterm labor that your team is uncovering, and such as microRNA or
Mhm.
microbiome
Mhm.
signatures, and how might these transform the prenatal care?
So I have been working with a team in BGI, which is a Chinese company looking at cell-free RNA. And cell-free RNA can be used to predict quite reliably the onset of different types of preterm labor. So paper is in publication or preparation for publication. So hopefully you'll see soon that yes, the cell-free RNA is a useful way to predict types of preterm birth. And the implication of different mechanisms. The problem, of course, that we're going to have is that we're going to have to find specific treatments for each mechanism to stop the preterm labor in the people we have said, okay, you're high risk of preterm birth, and it seems to be related to this mechanism. What are we going to do about it? That's the next question. So yes, finding a marker is one thing.
Mm.
What are you going to do about it is another thing. In terms of microbiome, that work has been done at Hammersmith Hospital in the IRDB with Professor Phil Bennett. And they have published recently a series of cases where they've actually changed the vaginal microbiome. And in their series of cases, they reduce the risk of preterm birth. So that in itself is very interesting. If you're found to have a hostile microbiome and then you try and change it, that seems as though you're going to reduce the risk of preterm birth. But in a sense, that's one cause of
Yeah.
preterm birth. And there are myriad causes. So I think watch this space. More and more information is coming out, and hopefully we will be there. We will be able to identify what's causing preterm birth and treat it. That's what the
Wonderful.
great hope is.
Yes, very, very exciting, and we'll be looking forward to that. So, your goal includes establishing a dedicated center for preterm birth research at Chelsea in
Yeah.
Westminster. And can you share your vision for such a hub and the intended impact for that? Yeah.
So I don't know that it'll be at Chelsea and Westminster. We need a hub somewhere in the world which is going to be able to integrate all the data we're going to get from our big bump study.
Mm.
So we will be opening in Ludhiana shortly. We have the funding. We're just getting there. It's taking a lot of red tape to get there. And then we hope that we'll be opening in Ghana, for our second center. And we're raising funds in the United States to open a center in the United States. So we'll be getting a lot of information, both from the UK, other places in the world, and in Australia. I forgot, they'll kick me under the table when they see me for not saying Australia, forgive me, but Australia, where we'll put all the data together. And that is really having a bioinformatic hub, which will produce all this information, will allow us to produce that atlas, the cell atlas you talked about the human uterus, with the fetal membranes, placenta, maternal blood, fetal blood, understanding how they all work together. So it's more a bioinformatics center that we want to create. Quite where, I don't know yet, but we're going to advertise and we will see where the best place for it is.
I cannot help but express that I'm so pleased to hear the name Ludhiana. That is where I'm from. So I'm sure the local team there, Dr. Wonder and everybody, they are very
Yes.
scientifically very competent. They are the people at the top of the
Yeah.
basic science understanding. And I think you've chosen the right place and the people. So I'm very excited to know more about this space.
Watch
So,
it. Watch the space.
yes. So why do you see a center in sub-Saharan Africa, as you mentioned, as critical? And what are the unique, challenging, and insights that you would want to share about this?
Yeah, I mean, I think it's really understanding preterm birth across the globe. So
Hmm.
you can't just do it in the UK. Interestingly, in the UK, we do have a lot of people from all over the globe. So we will be able to see how does the environment change the effect of the genes. So it's going to be interesting, because we have a lot of people from the Punjab down not far from Chelsea Westminster and the West Middlesex, which we obviously have a hospital there as well. So we're hoping to see whether the change in environment, how does that influence preterm birth? But I think if we don't go to places where preterm birth is common, and in Ghana, it's about 18%. It's not as frequent as you find in the Punjab or some areas in the Punjab. We will not actually be able to find a treatment which works in the places where it needs to work. It's really that principle that we need to understand preterm birth across the globe to find treatments which work in each locality.
Wonderful. Very interesting. So the numbers are actually worse in Punjab as compared to Ghana's preterm rate. That's very interesting to know. See, this is sometimes when we talk on these podcasts and we deal with different people, we actually some things are totally unexpected, and we start looking at world with the different perspective that, unless we really know, we shouldn't assume things. Um, yeah, so I would now come to more of the slight non-medical side of it. So now that at this stage of your career, what are your other priorities? How do you see yourself in the near future? And after this, we'll just close the podcast. And thank you for your time already. Yeah.
Well, those who know me well know that I also work in heart disease in pregnancy. And we do a lot of work, particularly with my colleagues in Rotterdam, with Jolien Rouse-Hesselink. We're trying to understand how we should be managing mechanical heart valve in pregnancy. And again, we'll be doing a study, I hope, if we get the money from the BHF, in India, in South Africa, looking at the management of mechanical heart valve. And other questions like, is being pregnant good for your heart? I mean, it's a really interesting question. So understanding that, I have interest in diet, and as again, anybody who knows me will persuade. I am always telling everybody eat fish at least 3 times a week. It's good for your brain, good for your heart, good for everything. So I'm a sort of evangelical crusader about eating fish. And I do try to play golf. I'm very bad at golf, but if anybody wants me to come and play golf with them, I'm always willing. So,
Oh, wonderful, wonderful. So it has been always a very profound experience to see how you think, the depths that you reach when you think. And I think it is very important that some of your students are actually trying to copy that. So it's good to leave some imprints to other people when you have so much to give. So thank you very much for your time. I'm
my
really
great pleasure.
delighted that we could do this today. And I know you're very busy, but you've been very humble in accepting the invite. And thank you very much for your time.
Thank you for having me.
Thank you, everybody, for joining us today. And thank you.